lecturer at the School of Biological and Clinical Science at Queen Mary University of London, and I do use Drosophila for my research, but I also use Drosophila for another very important project, and that is a charity that I co-founded with two other women, Marta Vicente Crespo and Maria Dolores Martín Bermudo, both scientists, and the charity is called Dros Africa.
Okay, what is Dros Africa?
So Dros Africa is a charity now. It's a registered charity in England and Wales, but it started as a project with the idea of helping researchers in Africa to be able to do research. There are a lot of researchers there that have a position, they have a lab, but they have no way to run projects, and they try with mammalian system, it doesn't go anywhere. Drosophila can help them develop research, and we help them develop a Drosophila lab to achieve their research interests.
Okay, so why is this Drosophila better than other systems?
In the case of Africa, it's because it's very inexpensive, which is a major problem there. It requires very low infrastructure, no demanding infrastructure at all. It doesn't require any specialized animal house, any license, there's no ethic issues, and it's extremely easy to learn it, and you can get, millions of Drosophila for your PhD instead of 5 rats for your PhD, which is what sometimes happens in Africa.
So it's easier to use and cheaper.
It's easier to use, it's cheaper, it's very easy to learn, but it's also very impacting. You can model a lot of the human diseases in Drosophila.
Okay, so you go there, you teach them how to use Drosophila.
Yes.
And then you also mentor them,
Yes,
I guess, in
correct.
further steps. So it's working. So are these researchers now in Africa
Yes.
using Drosophila to
Correct.
do research?
So when we went to Africa, there was, to my knowledge, one Drosophila lab in South Africa. That was all.
Wow, just one.
One that we knew. And the first goal of Dros Africa was to create a community of scientists using Drosophila that are connected across Africa.
Okay, so they become independent.
They become independent, they would apply for their own grants. We help them set up the lab, we help them learn how to work with it, and we carry on mentoring them. And now I can say that this September we're going to have the first African Drosophila conference in Africa, for Africa, from Africa.
So you have a very good response and a lot of interests.
Yes.
Scientists that help you. The project is working. So what's in the future now? How will this project grow?
Yes, so we have achieved our first goal, which was to create a community of Drosophila scientists in Africa. And we now have our first African Drosophila conference in mid-September at the University of Ibadan in Nigeria. And there are scientists that are using Drosophila now coming from all over Africa. So that kind of network of Drosophila scientists is achieved. The next goal, we would like to set up research institutes that use accessible model systems for research.
Institutes from scratch?
From scratch.
Okay, well that's...
Just starting with Drosophila,
very ambitious.
Very ambitious, there are many years in front of us. It's starting with Drosophila, but there are other model systems that are better for Africa than mammalian model systems.
You mean like the mosquito?
C. elegans, zebrafish, mosquito, and so on, and help develop that. That would be great.
So all this revolves around flies. So can we have a look at some of the flies?
Of course, absolutely. Let's go to the fly lab.
Okay, so we're now in the fly lab. And I wanted to ask you, we know that flies are an inexpensive model system, they are easy to use, but Drosophila is known as being a very powerful model system. Could you explain us why?
Absolutely. So it has been a powerful genetic model system for over 100 years, and that is been used to really understand how the genome is organized, what genes are, what genes do. But it's also a very powerful system to model some aspects of human diseases. So there are labs working on cancer using Drosophila as a model system. There are labs working on neurodegeneration. So it is a powerful model system for, I would say, almost any question you're interested in. There are limitations. They are not exactly like us, but a huge amount of their genes are conserved.
What does that mean?
So it means that we are very close to a fly. We are very similar to a fly in our genome.
And can we have a look at the flies?
Absolutely.
And look at these genetic tools?
Yes. So these ones that we are looking at is a mutation in the eye, and I'll explain to you why we're using that. Normally, flies have red eyes, this one makes the eyes become white. And that by itself is not maybe a very exciting mutation for us because the eye system is different, but it is a way to detect the manipulation with an external trait in the body very efficiently.
Okay, so if the flies have white eyes, you know that the gene you're interested in is mutated.
Exactly, the other gene that you are.
So these are the genetic tools we talk about.
Exactly,
in flies. It's
exactly.
very easy to spot.
Exactly.
And I would like to know how you use flies in your research. So what exactly do you study in your lab?
Even though Drosophila is very powerful as a model for human diseases, we are not per se studying human diseases. So what we study is the cytoskeleton in cells, which is a very basic and essential structure within cells that is in all cell types, and it is in all animals, also in ours, and it's extremely conserved. And it is essential for basically any process that the cell has to go through, any tissue. So we study more how normal cells function and how the cytoskeleton, in this case, does its job for the cell to achieve what it has to achieve at a specific time in a specific place. And what is very important is to be able to look at it in vivo. So when
what does that mean?
It means that you look at the cytoskeleton's behavior exactly when it's alive, when it's doing it.
So in living cells?
In living cells. Okay. And that's very important. For that, you have, again, genetic tricks that allow you to label with fluorescent proteins that you can then use microscopes to look at. You label this cytoskeleton.
Okay, and this, what are we looking at exactly?
So here we're looking at a protein that binds microtubules, and that's labeled with fluorescent, and we can see how the microtubules are moving, they're flowing with the cytoplasm motion, and also you can see that there is a organization of the microtubule that is very precise. This other movie is a mutation in a gene that is important for this organization, and you can see now that they are misorganized, they're not moving the same way.
This is amazing. I have another question because you have your own lab.
Yes.
You teach as well.
Yes.
You run a charity.
Yes.
And you're also a mom.
Yes.
How do you juggle all of this?
It is a struggle, and I would say there are maybe, 4 points that are key for me to achieve this. One is that you need to love it. You need to love what you do. If you don't love what you do, I don't think it's worth doing. And life
because it's challenging.
It's challenging. Okay. It's challenging. It's all about solving challenges and organizing, and that's one thing. You need to love it. The second thing is you need to have a good supporting network. Hopefully your partner, that would be the best. And I would say to all women out there, think carefully about the partner because it will define your life quite importantly. And if you make a mistake, change partner and then get a better one. The third one, which I think is a very important point, is the matter of guilt.
Non-guilt.
Any guilt. You are not in the lab, you feel guilty about not being in the lab and not running the projects. If you are with your family, you feel you should be teaching. If you are teaching, you should be worried. There's guilt in so many levels.
Yeah. That guilt might not be intrinsic, it might be external. People might put pressure on you, you just have to learn to switch it off and not listen to those people.
I totally agree. Yeah. But even with the intrinsic guilt, you can handle it. You can learn to manage it, and I do that. And the fourth point is organization. You need to be very well organized, and you need to be kind of switched on all the time, really.
Yeah,
thinking. Multitask, but thinking, just use your brain. We have a lot of brain that we don't use. And I think those are the 4 points, but the partner is really key.
Okay, well, that's very good advice, I think.
Yeah, thank you.
Thank you so much, Isabel, for this.
Thank you for this. It's been a pleasure.
And good luck with all your projects.
Absolutely. Thank you very much.